In this video, we're going to go through all of these different methods that we can use to kill and destroy microbes. And a lot of it we're going to just kind of go through quickly because there's a lot of different methods, and I'll just kind of tell you some of the key things that you should know. So we're going to start with talking about the physical methods such as heat, cold, desiccation, radiation, and filtration. So starting with heat, you could have either moist heat. Moist heat is much more effective at killing microbes. It requires a lower temperature in a shorter period of time because you're combining the heat with steam and moisture. And what it will end up doing is coagulating, which means clumping proteins together and also denaturing them, and so that the microbe can no longer function. Its enzymes can't function any longer. There's also dry heat. This requires higher temperatures and longer periods of time. It's not as effective as moist heat. Dry heat will dehydrate the cell, altering the protein structure, not necessarily completely denaturing it, but altering it so it's no longer functional. Couple of terms real quick. A thermal death time. This is the shortest amount of time it takes to kill all the microbes in a certain sample at a specific temperature. And the thermal death point is the lowest temperature required to kill all the microbes in a specific sample in 10 minutes. So let's run through a couple examples of moist heat. The first is an autoclave. You might have seen one if you work in a hospital. They also sometimes have them in tattoo parlors to sterilize equipment. And also, of course, in microbiology labs, we have these. Autoclaves work under high temperature coupled with steam. And usually we will autoclave something at 15 psi, 121 degrees Celsius for 10 to 40 minutes. And the autoclave will ultimately denature the protein, destroy the membranes and the DNA of the microorganisms. Some materials are very sensitive, too sensitive to go in the autoclave. So there's some other moist heat methods. We could do tyndallization. So tyndallization is basically intermittent, on and off exposure to steam. 30 minutes to 60 minutes on, about 24 hours off, and then repeat for almost 3-day cycle. So for example, canned food can be tyndallized, some sensitive laboratory equipment or media. And also putting something in boiling water is a form of moist heat, but this does not sterilize. It will simply disinfect and destroy non-spore forming pathogens. Pasteurization is a form of moist heat that we use for a lot of food products like milk is pasteurized, certain juices may be pasteurized, even wine may be pasteurized. So pasteurization is basically a flash heating. So we're flash heating our liquid or whatever it is for about 72 degrees Celsius for 15 seconds. This is enough to kill most of the pathogens and a lot of the vegetative cells in the sample, but pasteurization does not sterilize. That's why your milk will go bad after about 10 days because there's still endospores present, and then they germinate and form vegetative cells. Ultrapasteurization is similar, but you're doing a slightly higher temperature for a shorter period of time. Now, some dry heat methods. There's incineration. This is when you basically put something straight into a hot coil or like into a flame. For example, when we in the lab use our loops and we put our loops in the flame, we're incinerating. You could have a dry oven. And these methods, basically, they're going to coagulate proteins, denature them, and the proteins are going to lose their function. Now there's these tables again that summarize everything I just talked about, and they're really great and useful to use for studying. Now let's talk about some effects of cold and also something called desiccation. I already told you cold is actually microbiostatic, which means it temporarily prevents microbes from growing or it slows down their growth, but it doesn't actually kill microbes. That's why when you put food in the fridge, the food will stay fresh longer, but it still will eventually go bad. Freezing actually also doesn't kill bacteria, it just temporarily stops them from growing. And it's really a bad idea when you take food out of the freezer, let it thaw. Now you're letting endospores reactivate, regerminate, and then putting food back in and out of the freezer is not a great thing to do. Once food is thawed, it should be eaten or thrown away. Desiccation is basically the removal of moisture, the removal of water from cells, because all cells need water. So when you remove a lot of it and dehydrate the cells, they may slow down their metabolism. It's not that effective though. A lot of cells can still grow even with minimal water. Similarly, lyophilization, which is freeze drying food. We might do this for certain meats and stuff like that. Also not always the most effective preservation method. The last physical method of control is radiation. And there's two types of radiation: ionizing radiation, such as X-rays we're going to talk about, and non-ionizing radiation, such as ultraviolet radiation that we're going to talk about. So in ionizing radiation, electrons are basically ejected from atoms and cells. So electrons basically leave the cell. An example of ionizing radiation are X-rays, gamma rays, and cathode rays. So you might have heard getting too many X-rays is not always the best idea because X-rays actually cause damage to DNA. Not only do they damage our DNA, but they can also damage the microbes' DNA. So what these types of radiation do is they actually cause double-stranded breaks in our DNA. So the DNA is made up of two strands, and both strands break with too much exposure to ionizing radiation. Non-ionizing radiation, such as UV light, what this does is also damage DNA, but UV light actually causes something called thymine dimers. So basically, if you have a DNA sequence made up of A, C, G, and T base pairs, if you have an area where there's two T's next to each other, the T's will actually bond together, forming something called a dimer, this double bond. And this dimer will distort the DNA. So the DNA becomes all distorted and it can't replicate properly. We use irradiation in a number of different things. We use it in food. Like you can see, this is an example of berries that were irradiated and ones that weren't. And it's very safe. It's not like you're being exposed to any radiation yourself. It's simply the fruits being exposed to help kill off microbes. We use irradiation in a lot of meats and ground beef, for example. We even use it a lot in the hospital, like we may use irradiation to sterilize medical equipment. Now, filtration is also a physical method of control. In filtration, you're not actually sterilizing or killing any microbes. You're simply removing them from a sample. So a lot of times we'll use filtration, for example, in our water. A lot of your water systems at home probably have a filter where the water will pass through and any microbes will get trapped on the surface of the filter. There's also what's called HEPA filters, which are used to filter air. For example, hospital rooms will have HEPA filters to try to filter the air. Our final method of killing microbes is to use chemicals, which can either be gases or liquids. Now, levels of chemical decontamination, there's 3 different levels, and it depends on what we need to decontaminate. So high-level germicides are sterilants. They must be able to kill both vegetative cells and endospores, everything. So for example, a syringe that you're actually using to penetrate skin and tissues, or like a scalpel that you're using in surgery, or like a medical implant that's going inside someone. These devices need to be very sterile because they're penetrating the mucous membranes and the tissues of the human. Something that's intermediate level means we need to kill most vegetative bacteria and viruses, but it's okay if there are maybe some endospores there. So for example, something that's coming in contact with mucous membranes, but it's not invasive. So for example, a colonoscope. And then a low-level tool would be something that's not penetrating any tissues. It's only being used on the surface or on the skin. So, for example, a blood pressure cuff that you may use, it could be disinfected using a low-level germicide. Now, there's a ton of different types of chemical agents we use in healthcare, and we choose it depending on what tool we're sterilizing or what the situation is. There's tons of different ones, and this table does a really nice job showing you the agent, its targets, its level, etc. I'm just going to run through them real quick and tell you any extra things that are important. So the first group are the halogens. An example of halogen is chlorine, such as bleach. Chlorine will actually denature proteins by disrupting disulfide bonds in the amino acids of the proteins. It's intermediate level. We use it a lot for wastewater treatment. Like the final step in water treatment is to chlorinate the water before it goes into our public drinking supplies. It's a disinfectant, so you can only use it on inanimate objects. It's not safe for human skin. Now, another example of a halogen is iodine. Now, iodine is safe for some human tissue. It's also intermediate level. It similarly will disrupt the disulfide bonds in proteins. So, for example, when someone gets a surgery, they'll often put povidone iodine. It's like that mustardy looking stuff. They'll put it over the site of the incision prior to the incision during surgery. Phenols, they actually disrupt the cell wall and cell membrane, and they disrupt proteins. They're actually pretty poisonous, Back in the day, like early 1900s, they used to use phenol as an antiseptic, but people were getting burns on their skin, so it's not safe for skin. It's not safe for ingestion, but it can be used on some inanimate objects. So for example, Lysol is a type of phenol, but actually you don't want to get that on your skin. There is some evidence showing that phenols are actually carcinogenic, so they're just not safe for human tissues. Chlorhexidine is a surfactant. Remember, that means that they disrupt the cell membrane. They can insert themselves and destroy the integrity of the cell membrane. It's a low to intermediate level. An example of chlorhexidine is Inhibiclens. Remember, we're testing that in lab this week, that pink stuff. There's also some mouthwashes that are types of chlorhexidines that they might prescribe if someone has like an oral infection. So this is a safe antiseptic. It's often used to clean certain wounds or for burns. Alcohols are intermediate level disinfectants, and sometimes they can be safe for human skin, like when we use alcohol in hand sanitizer, it can be an antiseptic. Now, alcohol is medium level effective, but the key is it needs to be slightly diluted. You need to have around 70% concentration in order to coagulate and destroy proteins of the microbe. 100% alcohol is not effective. It evaporates very quickly, so even on disinfectants, it'll evaporate. It doesn't effectively destroy the proteins of the cell. Hydrogen peroxide creates these things called reactive oxygen species or reactive hydroxyl free radicals. These damage the DNA and the proteins in the cell, especially good at killing anaerobic bacteria that don't have enzymes to help destroy these free radicals. Very low concentrations of hydrogen peroxide, I mean low, may be safe as an antiseptic for human skin. High concentrations of hydrogen peroxide, extremely dangerous. You do not want to touch high concentrations. In very high concentrations, it may even be sporicidal. By far, the most damaging chemicals are the aldehydes, which alkylate proteins and DNA. These can actually be sterilants. So for example, glutaraldehyde is a very high-level germicide. It can be used to destroy endospores on some heat-sensitive equipment. Similar to glutaraldehyde in structure is formaldehyde, which is more intermediate level. It's often used as a preservative, disinfectant. It is toxic, so you actually don't want to come in contact with formaldehyde. There's a couple gases to talk about that also can act as disinfectants or sterilants. Ethylene oxide is an extremely strong alkylating agent, extremely dangerous, but also a sterilant. It can kill even endospores. So a lot of hospitals may have these ethylene oxide chambers, kind of like an autoclave almost, like you put the equipment in, but instead of that high heat and pressure, it pumps in the ethylene oxide gas, and then there's a vacuum to extract the gas and safely get rid of it. So this might be used again for some heat-sensitive equipment that are certain plastics that may melt in the autoclave. Instead, we'll use this ethylene oxide chamber. Detergents and soaps are both types of surfactants. So for example, laundry detergent, it actually works to embed itself in the cell membrane and destroy the cells. There's also a group called the quaternary ammonia compounds, or quats, which are a type of detergent. Washing your hands with soap in and of itself, soap is not the best disinfectant. It's very weak. It's actually more like the mechanical action when you're scrubbing your hands and brushing away the bacteria. That's really what gets the bacteria off your hands. And finally, heavy metals. Now, some examples of heavy metals may be things like mercury, zinc, silver. There's actually a burn cream called silver sulfadiazine or silver nitrate that they'll put over burns and act as a disinfectant. Heavy metals have what we call oligodynamic action. This means they work even in very, very small amounts. There's even some like algae killer. If you have like a lake that's contaminated with algae, they might use some heavy metals to try to kill off the algae since it may be safe to put in the lake. These are very low level though. They're mostly used for like skin or burn ointments.